Hey everyone, this is Dr. Jyoti Bala. In this session, I will be showing you a very useful tool which is online laboratory calculator. Especially many of the young student and researchers face a lot of trouble, especially because we have to perform a lot of experiments and conversion related to our experiment. So this session will be useful for both biologists and chemists who are dealing with DNA, RNA and protein at daily purpose. So a lot of different experimentation and conversion you have to perform. So for that purpose, you can utilize this online calculator. So I will be showing you one practical demo also here. If you haven't used this tool earlier, this is Zbio calculator. It is an online calculator. It's very simple and it can be utilized for many purposes if you are from biotech, biology and chemistry background, especially from research sector. Then you might be doing a lot of experimentation where you have to convert your DNA sense strand to antisense. You have to calculate and get the ideas about protein, molecular weight, its PI and all these things. And if suppose you are working on radioactivity also, then you have to perform scintillation related experiments and all these things. So maybe you are doing spectrophotometry related experiments and all these things for your purifications and concentration calculations. Then you can easily utilize this tool. It's very useful and it's very simple also. So just use this URL and come to this page first. I will be putting this URL in my description section also. So for your nucleic acid, for your protein, for your microbiology related growth calculations, radioactivity calculation, you can very easily utilize this tool. So let's do one demo. I have few RNA and DNA and protein sequences with me. So I will be showing you, let's go to the page first and utilize those samples and we'll do one demo for you. So this is the online calculator, Zbio. By using the URL which I have shown you in my previous slides, you may reach to the page first. I will put the URL in the description section also. So as I mentioned, for your biology, for bio IT and for chemical and chemistry related conversion and utilization also, you can use this tool very easily. It's very simple and very useful. So in a laboratory, we do a lot of tasks related to nucleic acid and protein. Like sometimes we have to read our sequence, oligonucleotide. We have to convert the lower cases to upper cases. Sometimes we have to convert the complementary strand, the sense strand. We have to get the ideas about 5' to 3', and then we have to sometimes convert them into the complementary strand, depending on what kind of projects we are handling. Maybe you are doing PCRs and cloning. So in my sequence massager tutorial also, I have given you a bit of idea about those tasks, but that tool is only related for your nucleic acid. Here you can do certain analysis and conversion related to protein also, microbiology also, and daily kind of experiments which we do in the laboratory. So if you have already done some task in the laboratory, you have the idea that many a time we have to convert these oligonucleotide, we have to calculate the things related to spectrophotometry, about the getting the ideas about the quality of proteins and nucleic acid, and then you have to run centrifuge. Depending on the machine, sometimes it will run in RPM and sometime in kg on. So you have to convert all these things, and then molarity calculations, you have to convert weight to the molar quantity. So we will convert all these things here. So for daily purpose in the laboratory, it will make your things very easier, especially many of biologists are bit phobic to mathematics and computer. So it is going to help you while performing a simple daily uses of your calculations related to your experiments. So see here in the nucleic acid and amino acid sequence, you may go to this sequence utility. And here you can see you just have to paste your samples. If you are handling nucleic acid, DNA or RNA, then you have to paste it. If sometime your sample is in lowercase, you want to convert them in uppercase, then you can do those tasks. And for conversion purpose also, if you want to get the ideas about the complementary stands, 5 dash to 3 dash, 3 dash to 5 dash, if you want to get the ideas about the double standards, then you can do these things. Let me show you if I have some samples. So see, these are few of my DNA samples. So let's take this. I'm pasting here. So right now these are in uppercase. If suppose I want to convert them into lower thing, then see, we got the our nucleic acid oligonucleotide is around 218 long, and here it is in lowercase now. Similarly, if you have your sample in lowercase, you can convert to the uppercase also. If I want to convert them 5 dash or 3 dash, then also we will get this sequence accordingly. If I want to get the ideas about the double standard, I have to make them double standard, then also these things can be done. Many a time you are handling a lot of samples and you have to read it loud because we have to write it down. So these things you can paste here and you can read it loud, because sometimes we are manually designing the primer, or maybe we are getting the ideas about where's the deletion and addition. Then you can read your oligonucleotide strands over here. If you want to get the ideas about the 6-frame translation, like from nucleotide, what protein will be translated. I'm taking random sample right now, but you have to take in the consideration your gene and ORF, everything you have to consider when you are handling the actual samples. So based on that, from your nucleic acid, the gene, you can get the ideas about what protein will be translated. So right now I'm like for your display purpose, I have taken the random thing, but for your actual gene, you have to take the right gene sequences. Similarly, for reverse translation of amino acid, from amino acid, if you want the reverse translation information, then you have to paste your protein sequences and then you get the ideas about that. Similarly, other mutation task and protein sequence analysis also you can do. So for protein analysis purpose, we mostly use ProtTram, like XPC is a very wonderful database and resource. So we mostly use ProtTram for those kind of analysis, but similar tasks can be performed here also. So if you will paste your protein, it will give you the ideas about your protein analysis also. So let's take some example of protein also. So I think I have few samples. So here you have to paste your protein and then calculate. So see, they're giving you example about your amino acid, the protein composition, this chemical formula, molecular weight in terms of Dalton, and PI values. You know, these values are very important when you are performing affinity chromatography, or you may be running the gel related to protein. So depending on these values, you decide the beads, the column, and your gel also. Similarly, if you are doing PCR related experiments and all these things, so you know, many a time we have to troubleshoot, we have to standardize these things. So here you have all these calculation options. So based on that, what kind of template, how much templates, what concentration you are using, what is your total reactions, based on that you can like put your values and you can do calculation, like how much template you require, how much DNTPs you require. So this will be also very important, because many a time you have to like standardize your own PCR. So this you can perform here. So many time you are using 25 microliter of reaction, sometimes 50 microliter. If you are using this old conventional PCR, there you might be using 500 ml. So, oh, sorry, 500 microliter. So depending on what kind of PCR you are running, you have to standardize these things. So what amount of DNTPs template you have to use, these calculation you can perform from here. And similarly, if you are using spectrophotometry for quantitation of your quality check of your DNA and RNA, then if you have done your OD, you have the values. So if you are handling double stranded DNA, single stranded DNA or RNA, depending on that, you have to choose those things, how much template you are using, what is the value of your OD. You can put all these things and you will get the ideas about the concentration and about your nucleic acid. And yes, in the laboratory, many a time someone is using double stranded. I myself has for aptamer project, I have used double stranded, single stranded, and RNA also, and synthetic oligos also. So for me, it was always good because the values get changed. So you get the idea about the OD, put all these things and get the ideas, and you can easily calculate the concentrations. Similarly, if you are handling this microbial things and bacteria, you have to calculate the time for like calculations about the growth related experimentation. So you can do these tasks, especially if you are from microbiology and biochemistry, and if you have to perform these tasks related to protein purification and all these things. So based on your bacterial culture, OD, you can do these tasks to get the ideas about the bacterial growth. So you just have to plot here the values and you can calculate easily. Similarly, you have other options also, like for centrifuge purpose, depending on which machine you are using. So we have different values for that, and you have to sometimes convert the, if you are using in RPM or centrifuge in kg, so you can do these conversion easily here. And many of you might be using radioactivity also, like as I said, for my aptamer and RN projects, many a time I have utilized and I have performed in vitro transcription. So for that purpose, I have used this radio labeled metals and all these things. So for radioactivity things, we have to perform scintillation count, and sometimes we have to run the radio labeled gel. So for that purpose, you have to get the ideas and you have to perform the calculation in different units. So all these things we can perform here. And similarly, a very important part will be your molarity related thing. So how much is the mole and molarity related to your nucleic acid or single stranded or RNA or DNA you are using. So you can like first select your samples, how much you are using, and then you can calculate how many molecules are present. So if I'm using this much of 1 microgram, so how many moles are present, and total how many molecules are present. Because if you are running very specific real time and PCRs and aptamer kind of project, then how many molecules of your nucleotide or oligonucleotide you are using is very specific. So we have to at molecular level calculate how many molecules are interacting with different macromolecule or different ligands and all those things. So all these calculations you can perform here. You just have to give the ideas which molecules, which oligonucleotide you are using, like double stranded, single stranded, RNA, or synthetic oligo, how much you are using. So based on that, they will calculate and give the ideas. So for this molar quantity conversion, you can use. And similarly, not only for nucleic acid, for protein purpose also you can utilize. Then you have to give the ideas about the protein, what is the molecular weight, like in last analysis we have showed you, based on the amino acid sequence, you can get the ideas about the molecular weight of your protein. So you have to put this thing, how much amount you are using, and based on that you can convert these things. And similarly, this nucleic acid thing we have shown you. So other calculation in laboratory, very like molar quantity to molar concentration we have to. So this conversion very easily you can convert, especially in laboratory primers. Sometimes we are using in millimole and then we have to convert in millimolar and all these things. So for this purpose, very easily you can convert. All these values are given, like whether you are using in millimole, micromole, nano, pico, femto. So these things in your molar concentration, depending on how much you are using, in what amount, like in 500 microliter or 200 microliter. So what will be the molar concentration, final molar concentration it will calculate. Okay. So I hope you will come to this page and try this calculator. It is very easy, and if you are a student or young researcher who are doing these kind of tasks, I hope this will be useful for you. Give it a try. Hope the session was relevant for you. If you have liked the session, I would request you to like, subscribe, and share the information. If you have any queries and if you want to learn these things, you can approach me and drop me message also. And do share these information among your scientific endeavor also. Thank you so much.